Dear students, today we are going to discuss about a new topic, that is on prevention and control of viral diseases. This is the topic of your module 5 of your course BMB202. First, we should discuss about the outcomes after completion of this particular module. What should you learn from this module, and how you people should be able to evaluate the measures that have been developed to control virus particles, and the conceptual basis of the control strategies. What are the strategies already taken to combat different viral diseases? The second thing, you people should be able to know the mechanisms of host immune responses to viral infections, the mode of action of different antiviral drugs or antiviral agents. And the third one is you are able to explain vaccine strategies and the mechanism of the vaccines, how the vaccines are developing immunity in our body. This slide will explain several important targets for antiviral therapy. So antiviral therapy can be of 4 different types. Our immune system, both types, innate immunity as well as acquired immunity, can combat against the viral infection. The second one is some antiviral drugs that may be, mostly they are synthetic molecules. Third one is interferons, and the fourth one is vaccine. So here, through this picture, I want to tell you what are the target site that we can focus on to target the virus particle, to hinder the entry of the virus particle, to inhibit the entry of the virus particle. In this red line, you can see what are the target sites, and here I have listed 6 target sites where we can focus on for the development of antiviral therapy. What are the sites? The first one is the entry of the virus. So if we can block the entry of the virus, then definitely human system, the cells are safe from the virus particle. So if the virus particle can succeed to enter into the host cell, mostly the virus particle, mostly the animal viruses, they enter into the host cell via receptor mediated, different types of receptor mediated endocytosis. In our third module, we have discussed about what are the types of receptor mediated endocytosis. So if the virus has entered into the host cell cytoplasm, but it's within the endosome, if we can block the release of the nucleic acid, it may be RNA, it may be DNA, if we can block the release of the genome or release of the nucleic acid of the virus particle from the endosome, then we can combat the infection of virus. And if the virus can succeed to come out from the endosome and can release its genetic material to the cytoplasm, or else it can be able to transport its genetic material to the nucleus of the host cell, then we have to develop a new strategy, because the virus will then either integrate to the host cell genome or else will go for subsequent replication. So we can block these two steps. Either we can block with some antiviral drug or with the other means of therapy. We can block the replication. We can use some inhibitor that can block the replication. We can use some inhibitor that can block the integration of the viral DNA or proviral DNA, proviral genome with the genomic DNA of the host cell. If again the virus particle can succeed to integrate to the host cell DNA and can succeed for replication, then we can try to prevent the virus particle, to inhibit the virus particle to further carry on, means we can block translation or protein synthesis. Because by the process of translation, the virus particle is actually synthesizing both the structural as well as the non-structural proteins, mostly the late proteins or the middle proteins. And these proteins will form, in few viruses function as polyprotein. They assemble to prepare the capsid. And if we can succeed to inhibit the synthesis of protein, or if we can succeed to block the translation process by using some translational inhibitor, then definitely there will be no formation of protein, no formation of capsid. And also we can use some inhibitor that can block assembly of proteins or polyprotein synthesis. And the last target site is the release inhibitor. So there are some proteins that are inducing the progeny virus particle, not the virus particle itself. It's the nucleocapsid to fuse with the cell membrane of the host cell. So if we can block this fusion protein, so they will not function, they will not carry or allow the nucleocapsid to fuse with the cell membrane to prepare their envelope and come out from the virus, from the host cell by the process called budding. Then again, we can get success, or we can combat the virus infection. So these are the 4 ways to combat viruses. Our immune system can combat antiviral therapy, interferon therapy, and via vaccination. Now, this slide tells you the type of viruses treatable with antiviral agents. So these are the listed viruses can be treatable with antiviral agents: herpes simplex virus, variola zoster virus, cytomegalovirus, human immunodeficiency virus, influenza virus, Ross sarcoma virus, 5 different types of hepatitis, human papilloma virus, and picornavirus. So first we will start with antiviral agents. What are the approach to antiviral chemotherapy? So the first approach is adsorption, penetration, and uncoating. To date, little is known about the specific reaction involved during these processes. So few antiviral agent synthetic molecules are available, like amantadine, rimantadine, that can be used against influenza A virus. with the following action. What are the actions they are actually doing? They are actually inhibiting absorption, penetration, and uncoating of the virus, influenza A virus. And now research is going on with this type of synthetic molecules over HIV, rhinovirus, Epstein-Barr virus, and the data shows that particular type of molecules that can combat absorption, penetration, and uncoating of HIV, rhino, or Epstein-Barr viruses. But the trial is going on. The second one is replication of viral nucleic acids. Means they can attack the enzymes, the synthetic molecules that are called as antiviral drugs or antiviral agents. They can attack the enzymes which actually catalyzes replication of the viral nucleic acid, not the replication of the host cell DNA. So all RNA viruses like pox virus, herpes virus, adenovirus, the work is going on. Next one is integration of viral genomes into cellular genomes. So as part of multiplication cycle, retroviruses. So in our previous lecture, we have discussed about how raw sarcomavirus is integrating with the host cell genomic DNA by the function of the enzyme integrase. So if we can block the function of the enzyme integrase, these retroviruses will not succeed to integrate into the host cell DNA. So we can prevent tumorigenesis by using this antiviral chemotherapy. And we can get success on the viruses like papovirus, herpes virus, Ross sarcoma virus that are actually responsible for tumorigenesis. The fourth approach is synthesis of viral messenger RNAs. So viral messenger RNA is a very, very important molecule in case of RNA viruses, in case of DNA viruses also, because they are actually guiding the DNA or the RNA, because it's an RNA-dependent RNA polymerase that is actually responsible for the synthesis of mRNA. So if we can block the function of this virus-encoded RNA polymerases and also restrict the capping of enzymes, then we can get success, because this virus-encoded RNA polymerase will function for the replication of the RNA, and the capping enzymes are protecting the viral RNA or the viral genome from the other type of nucleases present in from the nucleases that are present in the host cell. So they are protecting their genetic material from the host cell nucleases so that they can retain the structure of their genetic material via capping. And if we can target this virus-encoded RNA polymerase and the capping enzymes, and if we can get success to block the functions of these two enzymes, then viral messenger RNA synthesis can be blocked. And the fifth approach is synthesis of viral proteins. Here, viral mRNA, in the next step, viral mRNA in association with ribosomes, will synthesize different structural and non-structural proteins via translation process. And this translation is different from the host mRNA translation. So it is easy to target this viral mRNA, not the host mRNA. So it will bind, the antiviral drug will only bind with the viral mRNA, will inhibit viral mRNA translation, not the host mRNA translation, so that it can block the synthesis of protein or polyprotein. The sixth approach is capsid-targeted viral inactivation. This is a very new approach. Here, the scientists are targeting mostly the fusion proteins that are actually responsible for polyprotein synthesis or capsid synthesis and assembly. So if we can get success to synthesize some molecules that actually bind with proteins that is synthesized by the viral mRNA, they can inhibit fusion of the protein subunit to prepare the polyprotein or to form the capsid, viral capsid. And if the viral capsid will not synthesize, so no progeny virus will develop. So this technique or this technology can be used for the treatment of retroviruses, flavivirus, partner virus, etc., etc.